So let's talk about the final stage of aerobic respiration, and that is oxidative phosphorylation. Now, oxidative phosphorylation is composed of two separate processes. The first being the electron transport chain, and that's where we're finally going to use those NADHs and FADH2s that were made during glycolysis, the oxidation of pyruvate, and the citric acid cycle. And the second process is chemiosmosis, that is, using a proton gradient to perform cellular work. So, if we look at the membrane, the inner membrane of the mitochondria, here we have the matrix. Now here we have the intermembrane space. This is the inner membrane. What we will find here are a collection of proteins. Now as I move from left to right, what I'll find is the electronegativity for these proteins increases, meaning this protein here is more electronegative than this one here more electronegative than this one. The NADHs and FADH2s that were produced during the other processes will now be oxidized as they donate their electrons to the electron transport chain, causing them to become again NAD plus and FAD. So the total number of NADHs I input to the electron transport chain equals the number of NAD pluses produced during it, and the number of FADH2s I put into the electron transport chain also produces an equal number of FADs at the end. Okay, so as they donate their electrons are then passed from protein to protein to protein. And at the very end of the electron transport chain, those electrons combine with oxygen, which is the final electron acceptor, and that combines with protons to form a molecule of water. Now, as the electrons are being passed through the chain, their energy is slowly decreasing, but also what's happening is as they pass through each of the proteins in the electron transport chain, they result in the pumping of protons across the mitochondrial membrane. And this pumping of protons across the membrane results in us forming a proton gradient. Now, if we just look at this gradient, from what I have drawn inside the matrix to what I have drawn in the intermembrane space, you'll note that there is a higher concentration of protons in the intermembrane space, meaning that if we're looking at diffusion, these protons will, if given the ability to, diffuse down their concentration gradient from the intermembrane space into the mitochondrial matrix. So, this first part here is the electron transport chain, or the ETC. It's the first process involved in oxidative phosphorylation, and it allows us to generate this proton gradient. The second process, chemiosmosis, utilizes a specialized enzyme that looks like this. This specialized enzyme is called ATP synthase. As its name implies, it's an enzyme used to synthesize ATP. And here's how it works. The protons move into an empty space in ATP synthase. And they occupy these regions, these cavities in the molecule. and they cause it to turn. As they turn it, protons will be released onto the other side of the mitochondrial inner membrane into the matrix, and as they cause it to spin, they also cause another region of the molecule to spin, and that motion, that movement, that proton motive force is adequate enough to generate molecules of ATP from ADP and an inorganic phosphate. So it is the use of the electron transport chain to generate a proton gradient, and this proton motive force, and the use of that proton motive force in a process we call chemiosmosis, that contribute collectively to the process we call oxidative phosphorylation. And this process, total per molecule of glucose, has the capacity to generate between 26 to 28 molecules of ATP. Again, for this to have occurred in any capacity, we have to have molecular oxygen present if it's aerobic respiration. And we have to have generated the NADH and the FADH2 in previous processes to provide the electrons for the ETC. So that's oxidative phosphorylation.